semi-permeable property of the cell membrane acts as a gatekeeper for determining what can enter and exit the cell. This protection from the external environment is vital for the survival of all animals. Cell membranes are composed of mainly two layers of phospholipid molecules, with phospholipids have a head which is hydrophilic, meaning they are water-loving, and a tail that is hydrophobic, meaning they are water-fearing. This attraction or repulsion to water is directly linked to the polarity of the molecule and the chemical structure of it. The heads of the phospholipid molecules are polar and are composed of many possibly positively charged groups paired with a phosphate group. The head and tail of the molecule are linked by an intermediate glycerol group. The two tails of the lipid are nonpolar, and each tail consists of a hydrocarbon chain derived from a fatty acid. The phospholipids use their semi-permeable membrane to be gatekeepers of the cell, permitting some molecules entry through simple diffusion. Likewise, proteins act as gatekeepers, permitting larger molecules entry into the cell. There are two main protein guards known as integral and peripheral proteins. Integral proteins are fully nestled in the membrane. They span the membrane. To remove them would be to deconstruct the membrane itself. Peripheral proteins, they are bonded non-covalently to membrane components and are at either the intracellular or extracellular side of the membrane. There are 5 major gatekeeper proteins, better known as functional membrane proteins. The first is a channel protein. It creates a direct path from the outside of the cell into the cell. It employs the concentration gradient for diffusion of larger molecules, such as sugar and other things. The second functional membrane protein is a transporter protein. The protein pump binds non-covalently with molecules or ions, moving them into the cell. The protein pump can either employ ATP, this is known as active transport, or the protein pump does not require ATP. This is known as facilitated diffusion. Either way, molecules enter the cell through the pump. The third functional membrane protein is an enzyme. Enzymes are proteins that catalyze and regulate all chemical reactions in an animal cell. Depicted are the covalent bonds formed and broken between an enzyme and its substrate. This allows the enzyme to carry out its roles. The fourth functional protein is a receptor protein. When this protein pump binds non-covalently with a specific molecule, a change in cell permeability and cell metabolism is initiated. This pump participates in signaling, and many receptor proteins react to and translate signals being received from the extracellular. The last functional membrane proteins are structural proteins. They are vital in the shape and structure of the cell. They not only attach to other proteins, they also anchor to cytoskeleton filament and create junctions with other cells. The bilayers are fluid. This is due to the individual molecules not being covalently bonded to one another, meaning the lipids and the proteins between them are in constant motion. Fluidity is dependent on temperature and chemical saturation. The cooler the temperature, the stiffer and less the fluid the membranes are. The inverse is true at higher temperatures. Think of butter melting in the heat and becoming stiff in the fridge. Recall the tails of the phospholipid molecules are composed of a chain of hydrocarbons. A saturated hydrocarbon contains no double bonds in the chain of hydrocarbons. The straight tails allow the heads of the molecules to be more tightly packed. This decreases the fluidity of the membrane. While an unsaturated hydrocarbon has double bonds within it, these double bonds create kinks or bends in the chain. The kink in the tails does not allow for the heads of the molecules to be tightly packed. This increases membrane fluidity.